Okay, let's get started. I apologize. Last Thursday, I was at a conference that followed by two seminars. We recorded the lecture, but I forgot to upload it. It should be uploaded now. So starting from last Thursday, we are in the last module about cancer genomics. We'll teach you a few new techniques, but also hopefully you can also see how to utilize some of the skills that you have learned in the first two modules. So if you were to look at the first lecture, in last Thursday's lecture, you will see that that lecture is mostly focused on DNA, mostly whole exome sequencing for mutation analysis. And so today we're going to talk about tumor subtyping. We mentioned that there are basically National Cancer Institute of the US started this TCGA project by this time about 10 years ago. By this time, they have profiled 33 different cancer types, 10,000 cancers. And they use a number of these different profiling techniques. Initially, they were using arrays. Recently, moving more and more into sequencing. With sequencing, you can now sequence DNA to compare the tumor with the normal. So in TCGA, initially people also tried the normal as the adjacent normal, but now people believe the best normal should be the blood, for the correct, like your normal genome. Because in case you take adjacent tissue and that one also have mutation, then you would not be able to call it. So you compare the tumor DNA with the blood DNA to look at the cancer normal difference. Copy number variation can be called directly from the whole exome sequencing. Well, we'll talk about that a little bit. But then there's gene expression. Early days, people did microarrays, but then they have RNA-seq. There's also work on DNA methylation. We'll talk a little bit. MicroRNA, this is like small RNA. RPPA is a type of protein microarray. So you have different protein probes on a microarray, and you flash the proteins onto the array to see which spots light up. That can tell you also which proteins are expressed high or low in the tumor samples. And they have data on maybe like 1,000 different proteins. And then there are some clinical data. So in the clinical data, we know the patient age, the gender, the diagnosis, the survival, and how they are being treated. Most of the tumor samples were taken before the patient were treated with a drug. But you know what they were treated with and how long they survived and things like that, okay? So different profiling type. In the last lecture, we'll talk about mutation analysis, but today we'll talk about expression. For expression, in the very early days of TCGA, they used expression microarrays, but then now it's all the samples have RNA-seq data. There are also microRNA and protein. In terms of analysis, we will focus more on the RNA expression because it's the most complete one and it's kind of very informative. So for data analysis, imagine you have RNA-seq. What do you do now that you have gone through the expression analysis lecture? If we have, by the way, in here, we have tumor and adjacent normal. You do not want to use the blood as your normal because that's a very different tissue from your, whether it's liver cancer or breast cancer, you want the adjacent, normal liver and normal breast to calculate differential expression. But what would you do very quickly?
You do differential expression between tumor and
yeah, so for RNA-seq, you would do read mapping, right? Either with STAR or Salmon. You do differential expression like DE-seq. Then you could look at some, whether there are shared differences. By the way, there is a resource called the GPA, Gene Expression Profiling Interactive Analysis. This one has all of TCGA data. You can enter the cancer type, look at it. It will tell you on each chromosome which genes are differentially expressed at whatever cutoff you give it. So you can interactively play with it. So this nice thing about TCGA is All the data are uniformly processed, batch effect removed. So TCGA did a really nice job in that for each tumor, they had one site. So first of all the tumors from different hospitals are all sent to NCI. Pathologists look at the tissue slides to ensure that the sample quality. And then for each cancer type, all the samples are sent to a single center for profiling. So there's like cancer normal altogether. So minimize the batch effect. Okay, so this is just for differential expression. This one might be a little bit kind of a more advanced. If you have time, you can explore this. If you have a big cohort of microarray samples or RNA-seq samples, you could look at which genes are co-regulated, and they can form these modules. So so you can identify gene modules based on whether they are co-expressed in all the tissue samples together. You might want to do this for the genes that are differentially expressed between cancer and normal, and see whether they always co-occur together. And maybe they are important for some important functional pathways. Yes, Jet.
How does that, so this is like comparing multiple different samples?
Many samples, yeah. So most of the TCGA, remember we say there are 33 cancer types, total 10,000 samples. Some are bigger cancer types. The biggest, I think, is breast cancer. You have over 1,000 samples. And so based on each, all the samples together, you can see, oh, this group of genes are always together, upregulated or downregulated. Then you might guess that they are co-regulated by some transcription factors, right? You could then do your gene regulation stuff, see, whether you can figure out what is regulating those genes and things like that. Yeah, so that's more advanced. In the very first module, we talk about Geneset enrichment analysis. This is to compare every tumor with like every differential expression. Say if you have a cancer normal pair, you could look at the differential expression, right? And then based on the ranks of all the differentially expressed genes, you can run Geneset enrichment to ask what pathways or signatures are enriched in those genes that are differentially expressed. In this case, for TCGA, they did not get all the tumor samples with a matched normal. Depends on the cancer type. You may or may not be able to get it. I think for some cancer types, like lung cancer or breast cancer, you can always get some adjacent normal. But imagine if you have brain cancer, after surgery, you do not want that doctor to take out a piece of your normal brain just as a control for your tumor, right? So the end result is for TCGA, for many cancer types, they might have 300 tumor, but they only have like 30 adjacent normal from those patients. And so we don't always have the correct differential expression for that pair. So recently a tool was developed, developed in 2013. It's called the single sample GSEA. So they were able to create you can select what the baseline is. For example, you can do a pan-cancer as normal or all the tissues as normal, or you can select all your immune cells as your normal, or at least as your comparison. So whenever you have a single sample, you can compare that sample with a selected background, and they can calculate the differential expression, then run the Geneset enrichment analysis to see, for example, say you have 10 different samples. Each sample can compare to, you can select whatever as the background to calculate differential expression to see whether that signature is enriched or not enriched. And you can also select what signatures you want to look at, because you can imagine if you look at all Geneset enrichment signatures, there will be maybe 20,000. So you can pre-select which one you want to look at. And for each sample, you will run the GSEA for that sample for this signature and give it a value. So if it's minus, it will be a low signature. If it's plus, it will be a high signature. Then you can see all these patients, for example, this might be one cancer type, and you compare the two group of patients, you can clearly see that the patient are separated in two different classes. one class have really high signatures of a number of these gene signatures. The other is kind of low. Okay, so you can run on a single sample the Geneset enrichment to see whether some pathways are enriched. Right, and probably the most important thing is to do tumor subtyping with expression. So in this case, and so this one is protein, but you can imagine based on, you can do this for expression. So each column here is one sample, and each row is an expression. You can do this with RNA, you can do it with microRNA, you can do it with protein level, you can do it with mutations. So right now, what people found is that when you use different data to do the clustering, you might get slightly different clustering result. And so a method was developed for consensus clustering. So the idea is if you randomly sample the genes, say you get 70% of the genes, you get 70% of the tumors, or you try one profiling platform, you try another profiling platform, you try different ways, and you ask which samples are still clustered together, regardless of what platform you use, what genes you use, they always seem to be together. Then you create a pretty tight cluster. There might be some patients which don't really cluster in any of this. Could be a, say, misdiagnosis, or this is just kind of an outlier, but at least you know which ones are close based on, say, for example, based on protein, based on DNA, based on RNA, they all seem to be pretty close to each other. Then that's the cluster you want to get. Yeah, so this is a particular clustering is based on protein levels. This is the RPPA, which is the protein expression level. You can see some differences. For example, compared to the histopathology, actually pathologies also give some clustering or diagnosis or differences. ER, PR, and HER2 is, for breast cancer, for example, a very important way for people to do subtyping is for pathologists to use tissue slides and stain the tissues to see whether the breast cancer case have a high protein level of estrogen receptor, progesterone receptor, and HER2 as another receptor. You can also use mRNA to do the subtyping. You can also look at mutation type for different genes, and so you can try these different ways. So this is one way of clustering this. You can see this is one cluster, another cluster, and so you can see here this protein cluster is cluster 1, and you can see in this cluster mostly estrogen receptor. So ER is estrogen receptor is pretty high, and this cluster 3, you can see based on the expression, you can also see that the HER2 expression is pretty high, and so on. So this actually give you different subtype of cancers. So traditionally, many years ago, people were treating cancer. For example, if you have brain cancer, it's only been seen by the brain cancer surgeons or oncologists. If you have breast cancer, it's only been seen by the breast cancer oncologists or surgeons. And traditionally, people also do subtyping based on stage. If you have stage 1, it means the tumor size is small. Maybe with tumor, like stage 2 will be kind of bigger tumor, and then you might have a metastatic to the lymph node, which is, like say breast cancer, the lymph node will be like right close to the site, but just starting to metastasize. But then you might have a distal metastatic site, which means that the breast cancer has been metastasized to like liver or lung, right? So that's the kind of the early way how people do subtyping. But recently, we are starting to see molecular subtyping based on mutations, based on RNA expression or protein expression. And this is because depending on the subtypes, we are seeing quite a difference in the treatment options and patient survival information. Okay, so for example, in terms of mutation, recently the National Cancer Institute start to run something called a match trial or basket trial. It means that if the patient have the same mutation, regardless of where the cancer appear, if they have the same mutation, maybe they should be treated by the same drug. And those patients with that mutation might respond better, right? So these are kind of more in the precision cancer medicine age. We will have more of that in the next lecture. But also, you will see that depending on the different subtypes, you will see a big difference in the patient survival. And so that gives us the, introduce this concept of this survival analysis.